MARCKS was part of a trial that we published just recently, and which we discovered to be differentially expressed between mutated and unmutated CLL cases. So it was kind of really surprising data, because there were a lot of expression gene sets published until that time. And here in our approach, we identified it by using a proteomic, a global proteomic approach, where we compared the proteomes of mutated, unmutated CLL cases, and identified that MARCKS is one of the most deregulated proteins between both groups. And then we looked a little deeper by using that known or that available gene expression data sets, and this we could confirm. First, we could confirm this finding, and then we observed that there are, especially in the unmutated CLL cases, two groups of patients, such with very low MARCKS expression and others with higher MARCKS expression, comparable to those observed in mutated CLL cases. And another finding of the trial that was very important, I think everybody kind of supposed it, but it wasn't shown in that kind of global picture, because we also performed phosphoproteomics and compared mutated, unmutated cases, and we could show that the phosphorylation grade is significantly higher among the whole proteome in unmutated CLL cases. And then we looked how this, if MARCKS and the phosphorylation and activation degree is somehow connected, and which role it might play. And when you look on the role of MARCKS, it's a protein that shuffles between the cytoplasm and the cell membrane, and when it's on the membrane, so bound to the membrane, it sequesters PIP2, which is really a very important molecule to drive AKT signaling and general activation of a cell. So when you have a high MARCKS expression, and MARCKS is not phosphorylated, it's just on the membrane, then the B-cell receptor signal is much weaker. So in our opinion, MARCKS is therefore a really a new fine-tuning element in the B-cell receptor signaling cascade. And in unmutated cases where we have only low MARCKS expression, or MARCKS is phosphorylated, so it's not bound to the membrane, and it does not bind to PIP2, the B-cell receptor signal is stronger, and we have a general higher phosphorylation degree in unmutated cases. And then we were kind of happy that we could collaborate with Adrian Wiesner's group, who already had data on BTK inhibitor from patients that were treated with BTK inhibitors and also responded and did not respond so well, both groups, to BTK inhibitors. And we could really confirm that MARCKS does play a role for leukocytosis, and it is also important for the response to BTK inhibitors. So once the patient relapsed on Acalabrutinib, we saw again a higher degree of MARCKS expression in that patient. So to sum it up, I think we could identify with MARCKS a novel fine-tuning element for BTK treatment, and an element that discriminates also in the group of unmutated CLL patients between maybe very good and medium responders. But I think this needs to be checked in further trials.